Hello, my name is Kelly Dawe. I'm here to explain or give you basically the final status report of the major project to sequence and assemble the NAM genomes. And we did this primarily to address a real need in the community, which was that to do comparative genomics like we frequently do for almost every kind of science we do in corn, we need to have truly comparable genomes of high quality. So together with multiple colleagues, we wrote a grant and got it funded over the last 4 years and completed this work, which I'll describe today. So most of you are aware that the NAM genomes were the founder inbreds used for the nested association mapping population, so-called NAM lines, were chosen by Ed Buckler and colleagues some years ago to represent most of the diversity in corn. And the initial idea, which was well executed and heavily used, is to cross at that time primary reference B73 with each of these different inbreds to create F2 and real populations, which were then used for quantitative mapping experiments. They were chosen to represent the diversity throughout the Americas, including the temperate lines, which include grown in the Corn Belt. It also includes sweet corn and popcorn, also a large collection of tropical lines. And you can see that that diversity also includes variation in genome size from somewhat smaller than B73 reference to somewhat larger than B73 reference. These differences in size tend to be because of differences in highly repeated regions. So we set about assembling these genomes using what is now a standard genome assembly process of using long PacBio reads. At the time we did this, the read length for PacBio was over about 15 kb, which is longer than most of the longest transposons in corn. So if your read length exceeds the major transposon size, then you can generally assemble over the major intergenic spaces, which has been before this a major hurdle. And so basically, with high depth coverage of PacBio with standard assemblers, you can get very good assembly of so-called sequence contigs, which are continuous regions that with some gaps where they ran into some area of low coverage or repeat structures that could not be resolved by the assembler. Then to take all these many contigs and put them in proper order, we also prepared bionano optical maps, which are large restriction map representations of the genome. And with the restriction maps, you can then order and orient the contigs properly on the chromosome. And this is then put together to create so-called hybrid scaffold, which is a proper representation in chromosome view of the genome of that inbred. So these are excellent assemblies. The N50, which is a measure of completeness, is about 50 times higher than what we had for version 4. B73, it's really good. There are very few gaps, but because there are very few gaps, you might be surprised when you run into one. I just wanted to describe the three types of gaps that are present in these assemblies. One is the so-called 100N gap. This is the worst kind. This is the type you expect to find where there's a gap where two things have been joined because we don't know what's between them. This generally happens in knobs and centromeres. And this scaffold gap is marked by 100 Ns. A different kind of gap, which is only found when you have hybrid scaffolding using bionano optical maps, are gaps where there's not 100N or 13N, and a gap of some apparently arbitrary size, which actually is a measure of the missing sequence provided by the optical map. Those are gaps, but we know how much is missing. And then there is another type, which is kind of an artifact of assembly with long reads in a highly repetitive genome like corn, where two contigs are assembling along and stop, but actually did overlap, but we didn't see that until we aligned them back to the optical map, which said, oh, these two are on top of each other. Then the assembler just puts them end to end, which creates a false duplication. And those are marked by 13N. So these are the major types of gaps, but the assemblies are outstanding. So then to annotate the genome, this sequence, the genome assembly is the easy part. The annotation is the hard part, and particularly the genes are the hard part. So we probably put more time into the annotation than we did any other step of the project. To start with, we had created cDNA libraries of 10 tissues of each inbred. Shown here, the tissues we used. This is a very good resource. These are very high quality cDNA libraries, high depth sequence. And we use that data as inputs for called evidence-based annotation, where you do reference guided transcript assembly. And then this becomes the basis for evidence-based annotation. This type of thing can only get you genes for which you have expression in one of the 10 tissues, and it doesn't do well when there's low coverage. So we also need to add to that called ab initio annotation, which is a machine learning type of annotation, which then predicts where other genes are that it may have missed because of the absence of evidence. And we had all these genomes that were assembled the same way. We could then compare them all to control for quality across all the inbreds to get a final annotation set. And we found that for all 26 inbreds, generally see about 40,000 annotated genes per inbred. But we know from other prior maize literature that although most corn lines have 40,000 genes, that doesn't mean that it's the same 40,000 genes in every inbred. So in fact, different lines differ with the number of genes or type of genes present, and this type of variation is called presence-absence variation for gene content. And so this comes from a couple of sources. An important source is the ongoing process of fractionation, which is a result of the most recent genome tetraploid event, which happened 5 to 12 million years ago for corn, and rapidly resulted in biased fractionation, massive genome downsizing, and chromosome rearrangement and centromere loss to recover something close to the original diploid size of the original parents that doubled to create the tetraploid. So the vast majority of this was done long before humans came along to domesticate corn, but our data allowed us to estimate to what extent it's complete and to what extent it's still going. And that is illustrated here for a very conservative set, and these numbers are not to be taken as some kind of numbers that you should remember. These are just to illustrate the general principles. So we took a conservative set of 16,000 genes that were still in syntenic arrangement with the sorghum, and again, they're called syntologs, and asked the question of this 16th set of 16,000 genes, what percentage are present only in one subgenome, that is effectively single copy, or how many of those are only one, and how many of those are present in both subgenomes or effectively 2 copy. And so the answer is something less than half are single copy, and something more than half are effectively 2 copies. And that doesn't mean that the 2 copies are identical. Sometimes they are identical, both being identical to sorghum, but sometimes one is identical to sorghum and one is different, but there's still 2 copies. And then there's another set, which we discovered because we had excellent assemblies of so many inbreds, which are single copy in some lines and 2 copy in other lines. And it depends on which line you take. These are so-called segregating fractionation. It's an important source of PAV in maize. So that's one source. The ongoing fractionation process creates PAV. Another source of presence-absence variation is simple segmental duplication, basically tandem duplication of genes. That's the type we know we see in corn a lot. And we've observed that 16,000 genes in corn were duplicated in at least one line. That's a very large number of duplicates. And then we also saw the creation of new genes, or I would say evolutionarily young genes, which are present only in corn. This is true for all species, but we were able to, with so many good assemblies, estimate it pretty well here in the NAM lines. I'm going to start off by talking about the ones that are highly conserved. So we expect a very important gene that's present in all green plants to be present by sequence alignment. That is in a very high phylostrata, and this is the so-called the Viridiplante phylostrata, where that gene in corn is present in all green plants. For a lesser conserved gene, we'd expect it to be present only in Poaceae, a lesser conserved gene only in Andropogony, and genes that are relatively new and only found in corn should be also present. We might expect them to be smaller genes, which they are. We might expect them to be lower expressed genes, which they are. And we might expect to be a higher error rate in this class, which it probably is true. However, at least half of the genes in this category are present only in corn. We have evidence for indicating that these are a real and common class of genes. So that's PAV comes from fractionation, PAV comes from duplication or gene birth in recent history. So then we also, with all this kind of data, we're able to estimate the total number of pangenes in corn. This kind of estimate is difficult to do. Our estimate is that in these 26 inbreds, there are about 103,000 pangenes, but about only 32,000 are present in all lines. So you can see this visualized here. We look at the number, so this is a gene, this is the number of genes present in only one inbred. So that's the one here. This is the number of genes present in only 2 inbreds. This is the number of genes present in only 3 inbreds, and so forth until you get out here to the number of genes present in all 26 inbreds or 25 or 24 inbreds. These are called core or near core. These are probably the most important corn genes. And so we can see this is once you understand the graph, it's illustrative that you have many genes. The majority of the genes, the majority of the 103 pan genes, 1,000 pan genes are not core, but are segregating either as dispensable and at some reasonable frequency, perhaps from PAV or gene birth or gene duplication. And a lot of them are unique only to one line. So you may want to see this information. We have provided this information through something called the pan-gene matrix, which can be found on Cyberse. And basically, I'm just showing you what the pan-gene matrix looks like. So you may have a gene, you look around and find it in another inbred. This shows a truly a core gene that I'm sure is present in all inbreds. And this shows another feature of the data, which is a slightly flaw, but I'd just like to illustrate it to you, which is for even important genes like this one, so not all inbreds, we have what's called a gene ID, which is an outcome of the annotation pipeline. In some cases, the annotation pipeline did not annotate even genes which are known to be present in all inbreds. So we knew that to be an error, so we went back and checked just to make sure that the gene was absent by doing blast alignment, and found in many cases that the gene was there, just wasn't annotated. In those cases, then the gene is indicated only by coordinates. If you want to find this information, you can find it in the pan-gene matrix. So in addition to genes, we also assembled or annotated all the transposons very excellently. I guess I said I feel very strongly that we have very good completeness over the transposon space and very good annotation. With the excellent read length and assembly and particularly the optical map, let us also get far into the most highly repetitive regions of the genome, the centromeres and the knobs. So we were able to assemble 88 of the 260 centromeres gaplessly. We were assembled a lot of the smaller knobs, but not the larger knobs because they're too repetitive. And you can see this also annotated on the browser and when you get there. But basically, this shows knob polymorphism. You can see the huge arrays of TR1, major not repeat, or 180, and they're present or absent in lines, as we know. And then the centromeres positions were annotated by SNH3 ChIP-seq, which is a histone variant, which identifies functional centromeres. And you can see that there is some variation in positional variation in centromeres across these lines, also annotated. So, a major reason for doing a pan-genome project is to identify things that you could not identify by SNPs. So, and these are called structural variations. These could be presence absence for genes, that can be large transposon insertions, deletions or duplications of any type that you can't necessarily detect using short read data. So we did identify SVs using a straightforward method, a well-used method of taking the reads from the non-assembled reads from one inbred and aligning it back to the reference to look for improper alignments. And with appropriate tools, you can identify conversions, duplications, and deletions this way. So among the SVs we found that about 7% are not marked by a SNP. So when I say marked by a SNP, that means there's a SNP in high LD with the SV that you might have found this location otherwise if you didn't have the SVs. But the 7% can be quite important, and that is shown here in the next slide, where this is GWAS analysis for northern leaf blight. I'm going to stop here start at the bottom here. This is the GWAS hits using SNPs for northern leaf blight across the NAM inbreds, and you can see these various hits across the genome. A notable one here on chromosome 8. And if you go to the SVs, which tend to be in high LD with the SNPs, you can see the same rough pattern, including the same one on chromosome 8. But for SVs, you can also see a good hit here on chromosome 10 that was not identified with the SNPs. So then finally for this project, we had genes, we had assemblies, genes annotated, we had SVs called, centromeres annotated, knobs annotated. We wanted to also annotate the epigenetic space. And the reason this is important is because we know that genes in maize are not that simple. So if this were an Arabidopsis gene, we might expect to have the transcribed sequence to be right next to the promoter, which is how it should be. But corn has so many transposons jumping around that frequently a transposon will jump right into the middle of the promoter and separate components of the promoter from the gene at far distances. These are often called enhancers, distal regulatory regions that then loop around during transcription and activate expression. So these are very important parts of the genome, and they're very hard to identify just by looking at sequence alone, because they frequently are derived from transposons themselves, or they have a nondescript sequence. You cannot find an enhancer just by looking at sequence. The most effective way to do that is by doing methyl-seq. So we did methyl-seq of all 26 inbreds and scored cytosine methylation in all 3 contexts. And the context we use to score unmethylated regions is the CHG methylation context. And the reason we chose CHG is because it's low in enhancers, low throughout the gene, including the gene body, where CG can sometimes be high due to gene body methylation. So these regions, we know from prior work, very precisely identify potential regulatory, active or regulatory regions of the genome, which we called UMRs. We also did finally ATAC-seq, which is an open chromatin assay for leaf tissue for all 26 lines. And this is ATAC-seq identifies active transcriptional regions of the genome, promoters or enhancers. And as you'd expect, the ATAC-seq peaks correlate very well with the UMR peaks. So all this data is available at MaizeGDB and in browser views such as this. So I'm just showing you one example. This shows the inbred Missouri 18W for the gene miniature seed, which is a classical maize gene. And this inbred has an Arabidopsis look to it. There's an open reading frame and a promoter, which is unaffected. However, in most inbreds, there's a large gypsy insertion right in the middle of this promoter region, which separated the promoter into 2 parts. So this is now an upstream region. Other inbreds has additional transposons inserted there. So this is the genesis of these widely separate distal regulatory regions from the gene. All this data on the underlying raw data can be found at MaizeGDB. So just to wrap this up by saying that the assemblies we have completed are very high quality assemblies. I'm sure we could be a little bit better over some of those gaps, but it's very little improvement that can be done on these assemblies. We have an excellent expression atlas. All that data is available for all to use. We have carried out annotation to the best of our ability. This is a very challenging part of genome assembly in corn, mainly because it's so repetitive, because we don't have complete information. And the transposons are also transcribed, but we have a very good representation that is reliable for the vast majority of genes. We've annotated the transposons very well. The repeats are assembled reasonably well, considering how difficult they are. SVs we have called using a relatively basic way of doing it, using read alignment. This will certainly improve in subsequent reports and show their value. And we have already well-characterized methylation data, and the raw data are all excellent, and ATAC-seq data, all very good data. And all this data can be found in browser format at MaizeGDB, or it can be found at the Cyverse data store. So to wrap it up, this is the complete list of authors and pictures of a lot of those people. It was a collaborative effort by myself, Matt Hufford, and Doreen, the primary PIs on the grant, and then Candy and Jonathan helped a great deal later on to become effective co-PIs. Main support was by this grant from the National Science Foundation, but we also had intensive support from MaizeGDB as the product went through, using funds from MaizeGDB, and multiple of our people were supported by the USDA. And with that, I will stop.